Sickness. Everyone gets sick sometime. Some of us recover quicker than others. Why? And when I fall sick, what is it that determines if I live or die? For thousands of years, people guessed at these questions. Some said that sickness was caused by the anger of the gods. Even those such as Hippocrates, the so-called father of medicine, who thought that sickness was a natural and material thing within the body, couldn't explain it. Medical knowledge was limited to sewing, bandaging, splinting, a few folk remedies and a host of practices that probably did more harm than good. The body was usually left to cure itself, and it often did so against the most amazing odds and in some strange ways. For example, the most surprising thing about the Black Death in the Middle Ages was not that so many died, but that 3/4 of people, living as they did in company with animals and dirt, survived. Another surprising thing was that a number of diseases could not be caught twice. For example, smallpox was particularly dreaded. It not only killed, survivors were often left hideously disfigured. Yet if you lived, you never caught it again. You were immune. The best thing was to catch a mild case of smallpox, whereby you might escape the horror, yet still get the immunity. Some people even deliberately infected themselves from the blisters of a mild case to do just this. Risky business. A doctor named Edward Jenner heard of a farmer's superstition that if you caught cowpox, you would be immune to smallpox. Cowpox was a similar kind of disease, but it infected cows and occasionally people. It wasn't fatal, and it left very few marks. In 1796, he tried a famous experiment. First, he inoculated an 8-year-old boy with fluid from a milkmaid's cowpox blister. Then, 2 months later, he tried the supreme test and infected the boy with the dreaded smallpox. But the boy didn't catch it. He was immune. The Latin name for cowpox was vaccinia, so Jenner called the technique vaccination. Meanwhile, the microscope had been invented. People were becoming aware that there were creatures in nature too small to be seen, an invisible world that teemed with life. There were strange creatures almost visible to the naked eye, the protozoa. But they were, in turn, monsters compared to even smaller forms, the bacteria. The bacteria went down in size to the limits of the microscope. They were the most primitive types and were seen to have no cell nucleus. Most cells, however, do have a nucleus and other structures in them, and it was slowly realized that all living things were built of cells. Then discovery was hastened by an unusual event. In the 1850s, the French wine industry hit trouble. Aging wine was going sour and becoming undrinkable. Sacre bleu! Millions of francs were being lost. The problem was given to a young scientist named Louis Pasteur. Pasteur used the microscope to discover that the sour wine contained a kind of yeast cell that wasn't found in the good wine. Yeast, of course, is put in wine to make it ferment. Pasteur suspected that this extra kind of yeast might be the problem. Since the souring did not start until after fermentation was finished, Pasteur suggested killing all the yeast right then by gentle heating. The desperate winemakers tried it and were not only delighted to find that it worked, the flavour was unaffected too. Then the French silk industry went down with silkworm disease. Again, Pasteur was called in, and again he resorted to the microscope. He saw microbes on the sick worms and the leaves on which they fed. He suggested destroying all the affected worms and starting again only with uninfected ones. Again, it worked. Pasteur then came up with the theory that disease is caused through infection by microorganisms. It was a theory whose time had come. By bringing in hygienic and sterile procedures and by seeking out and neutralising specific disease organisms, medicine turned a huge corner. Death rates fell dramatically. The two basic ways of dealing with infection are, first, to deny it entry to the human body by keeping things clean and antiseptic. And second, if the body is invaded by the invisible hordes, you can try to find some means which will kill the invaders, but not also kill the cells of the body. And this is not so easy, because the basic chemistry of all living things is very similar. Firstly, you have to identify the invader. The introduction of solid media on which bacteria would grow but not be able to drift away from each other made it much easier. The culture dishes still keep the name of their inventor, Julius Petri. The bacterial colonies are easily seen and tested. Many successful drug treatments were engineered. However, it wasn't long before an even smaller form of life than bacteria was discovered, a form which was quite immune to the drugs deadly to bacteria. Viruses are 1,000 times smaller. Even Pasteur suspected that there were living things beyond the reach of a normal microscope. It took the invention of the electron microscope to see them. However, whether viruses are truly living things is open to dispute. When certain viruses were separated out in pure form, they turned into crystals. Yet, if redissolved, they were just as infectious as before. It seemed as if viruses bridged a gap between living and non-living things. It turned out that viruses could not reproduce themselves outside of a host body. Outside, they are not living, they're just chemicals. But once inside, they could reproduce themselves and cause havoc. What sort of a chemical could do that? The answer was the same type of chemical at the heart of a normal cell nucleus, DNA. Your average virus was a single DNA molecule with a protein coat. Inside the body, the protein coat attached to a living cell opened a hole in the cell wall and let its DNA contents slip through. Like a raider let into a fortress, the virus DNA takes over the cell's chemistry and reproduces itself, usually causing the death of the cell in the process. Any organism invading the body, whether it's a virus, a bacterium, or perhaps a fungus, is called an antigen. When it's recognized as an intruder, the body tries to defend itself by making substances called antibodies. The molecules of an antibody are specifically designed to fit the molecules on the surface of an antigen like a lock and key and destroy it. It's a race for survival. Our immune system must find an antibody that works and make enough of it before the antigen overwhelms us. But if our system does win out, it seems that it keeps a copy of the successful antibody at the ready for any new invasion. And so we become immune to that particular disease. This is what had happened in the case of smallpox. Because cowpox was an almost identical antigen to the deadly smallpox, the same antibody worked on both. But with the cowpox, our system would not be overwhelmed. Cowpox thus acted as a vaccine for smallpox. But finding a vaccine for many other diseases is not easy. For example, gastroenteritis kills around 1 million children a year, especially in third world countries. About 20 years ago, a Melbourne team of doctors discovered that many cases were caused by a virus. But only now have they managed to grow a vaccine, and it's still being tested. Part of the team was Dr. Ruth Bishop.
We went looking for the cause of acute gastroenteritis in young children coming into hospital. No one had been able to find a cause, so we turned to the electron microscope to look, and we found fairly quickly a new virus. It was identified as being new by its shape, and it was subsequently called a rotavirus, rota meaning Latin for wheel, and we discovered that it actually caused 50% of acute gastroenteritis in young children coming into hospital. We discovered a similar rotavirus in newborn babies in one of the obstetric hospitals in Melbourne. But the interesting thing about this rotavirus was that it was not causing disease in the newborn babies. It was probably a representative of harmless virus, which are very common in nature, able to infect, unable to cause disease symptoms. And it was this rotavirus that we felt had possibilities as a vaccine. It has been a slow process. Human rotaviruses do not grow well in the laboratory, and so we spent a long time trying to grow it. Once we had sufficient grown in the laboratory, you have to test it in young children under very, very restricted conditions, and that is the process we are developing at the moment. So the rotavirus vaccine is going to be used in developed countries like Australia and the United States because it is still a disease that is expensive to treat, so it will save healthcare costs. But it is mainly aimed at saving lives in developing countries, and this is the major aim of vaccine development at the moment.
So, at last we understand something about what causes disease and how our bodies respond to infection. But there's still a lot that we don't know about ourselves, and a world of microscopic creatures we have only partly seen. The fight against disease is far from over.